So I'd like to say a special word of thanks to Trevor and also the ARS Movers and Shakers for organizing this wonderful symposium, and in fact the events that are part of this wonderful occasion. And of course, thanks to Joachim for being here as well. So much of our research is focused on the structure and function of metalloproteases, and specifically angiotensin-converting enzyme, and the design of novel inhibitors which potentially can be developed into drugs for the treatment of hypertension and cardiovascular disease. And so angiotensin-converting enzyme was discovered in the 1950s by Leonard Skeggs. And this was pretty much a serendipitous discovery, as indicated in this quote of Skeggs. And so he was working in the Cleveland Veterans Hospital and working on the compound angiotensin, which was actually called hypertensin back in the day because of its effect on blood pressure. And one day when he was working on the kidney prep, he decided to use saline instead of water for the dialysis of the prep. And when that came to doing the countercurrent distribution to separate the compound, they found that instead of it actually appearing in the middle of the countercurrent distribution, it appeared very early on. And so they were astonished by this, and so called the one compound angiotensin 1 and the other angiotensin 2. And shortly after that, angiotensin converting enzyme was discovered. And the reason why they were getting this second band was that ACE is actually activated by chloride. So when they used the sodium chloride for the dialysis, this activated ACE and it cleaved the inactive peptide to angiotensin 2. So you probably don't need too much introduction in terms of the burden of hypertension, but just one or two important factors. Firstly, there's an estimated 1.3 billion adults between the ages of 30 and 79 worldwide that have hypertension, and most of these are in low and middle income countries, with about 10%, 10 to 12% in sub-Saharan Africa. And an estimated 46% of adults with hypertension are actually unaware that they have the condition, and less than half of adults with hypertension are diagnosed and treated. And this is why hypertension is often referred to as the silent killer, because often patients only realize that they have high blood pressure when they present with a stroke or myocardial infarction. And approximately 1 in 5 adults with hypertension have it under control. And obviously, it's a major cause of premature death worldwide. And one of the global targets for non-communicable diseases is to reduce the prevalence of hypertension by 33% between 2010 and 2030. So this brings us to the renin-angiotensin system, or the RAS, which plays a critical role in blood pressure regulation. And you can see angiotensin-converting enzyme occupies a central position in this cascade and converts the inactive decapeptide angiotensin 1 to angiotensin 2, which mediates its effect on blood pressure via the AT1 and AT2 receptors, which have opposing effects on. Now, ACE also has an important effect on the kallikrein-kinin system, which is shown on the right-hand side, and it inactivates the vasodilator bradykinin to an inactive peptide. And since the early 80s, ACE inhibitors have been widely used for the treatment of hypertension, and there are about 17 of them that are commercially available. However, because of the increased levels of bradykinin, a lot of the ACE inhibitors have adverse effects, such as persistent cough, which affects 1 in 5 patients in some populations, and angioedema, which is less common, probably affects 0.1% of patients, but is potentially life-threatening. Now, this is probably a more comprehensive view of the RAS, with a lot of other angiotensin-like peptides shown in green, and the ACE2, which converts angiotensin 2 to angiotensin 1 to 7, which then interacts with the MAS receptor and forms the protective arm of the renin-angiotensin system. So ACE is a fairly promiscuous enzyme, and cleaves a whole lot of substrates apart from the celebrated angiotensin 1 and bradykinin. It cleaves these neuropeptides like neurotensin, as well as this small acetylated SDKP, which is an antifibrotic compound. And with all these, it has a preference for cleaving at the penultimate C-terminal bond, releasing a dipeptide, but it's also shown endopeptidase activity on substrates such as neoendorphin, substance P, GnRH, and the beta-amyloid peptide. And so because of this wide substrate specificity, it's not surprising that it has diverse biological functions ranging from hematopoiesis, inflammation, through to immune regulation, male fertility, and processing of the amyloid beta peptide. So this shows the domain structure of ACE. There's a somatic form shown over here, which is comprised of two very similar domains, an N and C domain, that are likely products of gene duplication, and have about a 66% sequence identity. Each of them has a catalytic site with this zinc binding motif, HEXXH, where X can be any other amino acid. And the protein is decorated with these glycans shown by these little lollipop structures with 10 N-linked sites on the N domain and 7 on the C domain. And then there's a smaller testis form, which is transcribed from the same gene over here, shown above, but there's a tissue-specific promoter in exon 13, which results in the expression of the testis form, which is important for male fertility. And although these domains are very similar, and the active sites, in fact, have about a 90% sequence similarity, they do have different substrate specificities. So, for example, angiotensin 1 is more specific for the C domain, and other peptides like the A-beta peptide and acetyl SDKP are very specific for the N domain. And at present, the ACE inhibitors that are on the market don't bind with any preference to the N or the C domain catalytic sites. So this shows the molecular structure of the C domain of ACE. This was published in 2003 with our collaborator Ravi Acharya at the University of Bath. And the work was done by a postdoc, Ramanathan Natesh. And you can see that the domain is comprised of these two subdomains shown in cyan and purple. And in the middle is a ligand, an ACE inhibitor, which is bound, the zinc in green, and the two chloride ions are shown in red over there and there. And if one looks at this, the representation below, one can see that the active site is buried deep inside the molecule. And then you have this lid-like structure over here comprised of the first three alpha helices, which, as I say, form a lid, and the substrate gains access through a small opening underneath that lid. And on the right-hand side, you can see the sort of canonical zinc binding with the two histidines and glutamate, which coordinate the zinc. And then there's also a water, which is polarized and actually forms the nucleophile, which attacks the carbonyl-carbon of the cis-al bond. So what we've done in terms of focusing on the design of new inhibitors is to develop ligands that are specific for the C domain of ACE, and this leaves the N domain free to nibble away at the bradykinin and reduce the levels of bradykinin, which cause the side effects. And thus, the hope is that these inhibitors would have a much improved side effect profile. So this is probably our most promising early lead compound, which we call lystrip because it's a derivative of the inhibitor lisinopril, where the proline is actually replaced with this tryptophan-like moiety. And here we can see a crystal structure which was solved by Jean Watermeyer together with Trevor's assistance, binding in the active site. And if we look over on the right-hand side, we can see the molecular basis for the C selectivity of this molecule. And that's because the tryptophan-like group interacts with this hydrophobic patch caused by valine 379 and valine 380, as well as the interaction of this indole nitrogen with D415. So this explained the sort of 260-fold C selectivity of this molecule very nicely. We then did a small proof of concept study in hypertensive mouse model. This was work done by Dylan Berger, postdoc in Rion Toys's group, and showed that with our compound lystrip, one was able to obtain a 15% decrease in blood pressure at week 1 and week 2, which was similar to that shown with lisinopril. So this showed that by inhibiting the C terminal domain, one was able to get a similar effect in terms of lowering blood pressure. But more importantly, if we look at the levels of bradykinin in these animals, we can see here with the lisinopril, because one's blocking both domains, one gets elevated levels of bradykinin, whereas with the C-selective inhibitor, one got a dramatic decrease in bradykinin levels. And then an approach in terms of treating blood pressure has often been to target two different pathways that affect blood pressure. And so this is something that we've done as well, targeting the renin-angiotensin system, as well as the natriuretic peptide system down here, where you get natriuretic peptides, which have effects on vasodilation and natriuresis, which is obviously beneficial. And so again, with working with Ria and Toys and the hypertensive mouse model, we looked at the co-administration of our C-selective inhibitor with the neprilysin inhibitor sacubitril, and showed functional effects in terms of decreases in blood pressure and improvement in cardiac function, structural effects in terms of remodeling and the amelioration of mechanical properties, and then more importantly, favorable angioedema correlates in terms of reducing vascular relaxation, preserving the metabolism of bradykinin, so decreasing levels of bradykinin, and then decreasing vascular permeability so that one was able to reduce the flow of fluid into the extravascular space, causing the angioedema. So this really brings us to the next step, which was solving the Cryo-EM structure of somatic ACE. And I feel like a bit of an imposter because I'm not a Cryo-EM expert, but the star of the show is Lizelle Libber, who's probably one of the most resilient and innovative colleagues that I've worked with, who single-handedly solved the structure together with Trevor and Jeremy's assistants. And we knew a lot about the individual domains of the molecule. We had several crystal structures, a lot of kinetic data, but there were some very important points that we didn't really understand in terms of the orientation of these two domains in the full-length structure. Whether there was, we knew that there's some cooperativity between the two domains based on kinetic data. And then dimerization, whether what exactly was involved in terms of the interaction between the two monomers. And finally, whether there was an opportunity for allosteric regulation. So this just shows the different constructs, the full-length construct at the top, and the N and C domain truncated constructs, which were used for all the crystal work that had been carried out to date. And then at the bottom, the full-length two-domain structure, which Lizelle used, which was truncated in the juxtamembrane region. And so the protein was expressed in CHO cells and purified using lysinoprylcephalose affinity chromatography. And you can see on the left-hand side here a very nice band, single band, with a sort of broadish smear, which is a result of the heavy glycosylation. And then the protein was also subjected to size exclusion chromatography, which yielded a single peak for the monomer. However, if we look at the non-denaturing gel on the right-hand side, we can see that there's a small band for the dimeric form of the protein. We couldn't isolate this on the size exclusion, and this is probably because of the dynamic relationship between the two in solution. So this shows the structure of the monomer, looking at it from the top and from the front and the back, and you can see the N and C domains in blue and mauve, with the glycans represented by these green bubbles on the surface of the protein. And then importantly is this interdomain link region shown in purple. And you can see on the right-hand side how that sort of wraps around the C-terminal part of the end domain of ACE. And then on the right-hand side, I'm not sure if this will... there's a video that's not. Can you hover on the right-hand side molecule? It just shows. There we go. The flexibility of this linker region here, which, as I say, wraps around the bottom of the end domain. And so, in order to have a closer look at the dynamics of the monomer and how this could relate to any possible cooperativity, Lazar carried out a 3D variable analysis, and that's shown on the left-hand side, as well as normal mode analysis on the right-hand side. And you can see that there's this bending motion because of the flexibility of the linker region, this breathing motion, which is shown in B over here, which is the result of hinging, intra-domain hinging, opening the sort of two subdomains of the N and C domains, and then at the bottom, this jumping motion, which is almost like a sort of the inter-domain linker acting as a spring, and the two domains moving in and out from the N-terminal lid regions. Yes, so this shows the, if you can just start that. So this shows the twisting or pivoting. This shows the bending quite nicely. And this shows the breathing. And then the last one shows that jumping motion of the two domains. There you go. Okay, the next interesting insight was that the end domain was shown in this open cleft conformation. So here are the two subdomains, and there's this channel with the active site, and which was in the open form. And previously, with all our crystal structures, they were shown in the closed conformation, as shown in the top right. And this was in the presence as well as the absence of ligand. And the main reason that the structure without ligand were found in this closed conformation was that usually there was a molecule from the crystallization buffer that was found bound in the active site. And only recently we were able to get a crystal structure of the end domain in the open conformation. And that's seen by this little bit of daylight over here between the two domains. And then in this video that Lazar made, this was actually in a previous study looking at the cooperativity of the end domain in terms of inhibitor binding. We see this hinging through region 2, which results in the sort of opening and closing of the molecule. And this helped us to understand the cooperativity of inhibitor binding. So the inhibitor binds in this region here, but distal to that, in the prime site, we see interactions between these yellow and red molecules, which resulted in a sort of synergy or cooperativity in terms of inhibitor binding. And so in terms of this opening and closing, looking at the structural data as well as molecular dynamics that have been carried out by Lee et al., we're able to propose this model where the open and closed forms exist in a dynamic equilibrium. The open form binds to the substrate, which results in a closure of the molecule. And then after hydrolysis and release of the product, it reverts to the open conformation. And then finally, looking at dimerization of ACE, earlier work that was carried out by Karen Holstedt and Ingrid Fleming showed that ACE inhibitor binding promoted dimerization of ACE, which led to phosphorylation via casein kinase 2 and signaling via c-Jun, which resulted in increased expression of ACE and cyclooxygenase 2. And this might explain why one sometimes gets ACE inhibitor resistance with patients who've been on ACE inhibitors for a prolonged period of time. And then on the right-hand side, we see work that our group carried out. This was by postdoc Albert Arbery, showing dimerization on the membrane using biofluorescence complementation, and then work carried out by our collaborators in Bath using small angle X-ray scattering, showing dimerization, but in solution. But because of the low resolution, it's very difficult to assign these different domains. And so to determine whether the interface was actually between the two N domain regions or the C domain. So this shows the dimer structure that Lizelle solved from the top and the front, and the two N domains were well resolved, but the C domain was only seen in the raw map. And if you just hover on the right-hand structure and play that video, you can see the large sort of swinging motion of the C domain, again, which is accommodated by this flexible linker region, allowing the dimerization. And then at the bottom right, we just see the fitting of the model into the density in helix 23. So here we see the dimer interface in the N domain with hydrogen bonding and also some hydrophobic interaction, and at the bottom here we see glycan interaction at the top over here, and at the bottom interaction between these three C-terminal loops, loop 1, 2, 3, which form an allosteric switch in the molecule. And they're shown a little bit more closely here. So the orange loop is the interdomain linker, and then loops 1 and 2 in yellow and purple actually extend into the active site where they interact with the substrate and form this substrate anchor. How many more minutes?
Got 4 minutes, including questions.
Including. Okay, so the last interesting point in the dimer was that if we zoom in the active site, we see that one doesn't actually get the canonical zinc binding. In the monomer, you see the histine 361, which coordinates the zinc, whereas in the dimer, that rotamer is actually flipped out of the active site. And then on the bottom right, we see that this lysine 489, which interacts with the C terminus of the substrate, also faces out of the active site. And this suggests that this might actually be in a sort of resting or inactive site, at least in the N domain. And then finally, Lizelle looked at the potential for allosteric regulation using cavity plus, and once she'd found these cavities, used core-site 2 to predict allosteric sites based on the hypothesis that the motion at the active site and the allosteric sites are highly correlated. And so here we see 5 of these sites in the end domain and 3 in the C domain. And we're now using those to design or to screen for allosteric inhibitors. I'm going to skip over this. And so what we did was to use site 4 in the C domain, which is in the active site channel, but distal to the binding site, and use this for virtual screening. And this work's been done together with a startup company in Melbourne, work done by Omi Duggat and Mark Agostino. And so I started with the zinc database of small molecules that have known PK and PD data. And these went under 2 phases of screening, come up with about 360,000 molecules that were then counter-screened with the active site at the active site of the C domain, the active site of the N domain, and the at the end domain equivalent region of the C domain active site. And then this yielded a smaller group of compounds which were re-screened using a high accuracy of sampling, and finally gave 100 compounds of the highest scoring compounds. And so this just shows the screening. Here's the site 4, here's the active site, and then the counter screening in the end domain with the active site and the similar site to the C domain allosteric site. And this yielded a few compounds that inhibited between 150 and 200 micromolar. That's with a 50% inhibition. So they weren't great inhibitors, but their synthesis is well known, and we're looking at modifying them so that we can increase the binding of some of these. Okay, I think I'm going to end there, and just to, I think I've mentioned most of the collaborators, thank the funders, and this is our group based at the IDM. Thanks very much.
Thank you, Ed. That was a wonderful talk, and I think you gave Giselle and her work
Well,
fantastic exposure. I hope you're there.